as the introduction said, I'm part of the Center for Children's Environmental Health at UC Davis, where we focus on autism. And today, I'm going to take you through some of the work that we're doing. I'll give you sort of an overview of what we are, who we are as a center, but we really are looking at the role of the environment, genetic interplay with that, with the environment, and the immune system as they relate to autism susceptibility. And we do this through population studies and through animal models. So we've got a large group of investigators all working at this from different expertise, but looking at how the environmental triggers diet and so diet intake and immune dysregulation affect brain development. And then we're also looking for biologic markers and sort of markers of the response to the environment, markers of that might distinguish populations, the typically developing from the autism population, and try to understand if those markers can tell us something about the pathology or the etiology of this disorder. And we're particularly also interested in, so we're working in both children and the gestational environment. And I think, as we've seen from the previous presentations, the gestational period is very important, and that carries on through the postnatal period. But we're looking at how the immune system interplay, and I'll explain how we're doing that and why we think the immune system might play a role in changing neurodevelopment and leading to autism, at least in some cases. And then I'm going to touch on some work that we've done with maternal autoantibodies that recognize proteins that are highly expressed in the developing brain in autism, and that these antibodies are very specific for autism, and how their potential role in what they're doing in the brain. But I think to begin with, and when it's funny when you talk about 99, back in 2000, 2001, when we first started looking at this, I would have to stand up and say why I'm looking at the immune system in autism. And it was there was such a push to try to get past that and sort of past the thinking that those were two completely separate systems. And I think we've finally breached the wall on that. I don't get that in reviews of my papers anymore. It's like, we don't know that the immune system has anything to do with autism. Well, that's the point of the paper. But, sort of, you have to kind of that's why we submitted the paper. But I think, we now know that neurons produce cytokines. They have receptors for cytokines. This interplay is constant, not just during neurodevelopment, where it's very important, and as I'm going to talk about, but on immune cells have receptors for neurotrophic factors. As they respond to leptin, they produce leptin, as a matter of fact. And so there's a lot of what we didn't know now that we know. And so we can think about it as a whole system versus these two really separate compartments. So there's bidirectional communication. It's not one way. It's both directions. If you think about it, when you're sick, the fever is generated right from the response. Your body knows that you have the flu. Your brain doesn't understand that. I mean, your brain gets that signal from the periphery that you need to sleep and you feel really bad and you now have a fever. So all of that is actually driven from the periphery. And that an altered immune response, especially during gestation, may impact neurodevelopment. And that's a lot of the work that we're doing is if you change that equilibrium, especially during that very susceptible period of neurodevelopment, can you change the course of neurodevelopment? And what alters both? So we've looked in the context of environmental exposures a lot at neurotoxicity, right? And we've looked separately at immunotoxicity and some of the, especially data in mercury, very immunotoxic and neurotoxic. But now we're trying to marry those two systems. So now we know, so the exposures that happen in the periphery and what might happen in the maternal, maternal system in the periphery, how does that impact the brain? And do you have this concomitant exposure in the brain? So, they're not happening independently either, necessarily. So the etiology of autism is not well understood. Genetics, that was a great slide this morning. Thanks, Bruce, for it's like genetics research and everything else in autism. And so we sort of are constantly pushing against that wall as well. But the marriage of those two, genetics drives everything, obviously. We all know that, right? That's how we're built. But it's not, we need to look at those other factors, and we need to look at the interplay of those factors. So definitely environmental factors and the immune system, which is linking those two as well, or maybe perhaps as a marker or as a sort of a litmus test of what's going on as well in those systems. And then how the nervous system is sort of those, that interplay can affect the nervous system in general. In genetics, we know there's a number of syndromic causes of autism, or where syndromic genetic disorders where autism may be 30, 40, 50% in that population. The CMV is genetic susceptibility. I think I'm more particularly interested in the susceptibility part, because I think, and which is harder to answer, it's a harder question to answer, but I think that is really what we should be looking at. Environmental factors, there's a number of studies, I don't have to go through that with this crowd, but of environmental factors that we're looking at, and probably some unknowns that we haven't even contemplated yet. I think stress is a great environmental factor that we should be looking at. And then immune dysregulation, where that might lead to immune dysregulation both during gestation and immune dysregulation in the offspring. And knowing that the maternal immune system actually drives development of the offspring's immune system. So perturbation and dysregulation during development of the immune system has lifelong impact on the immune system of the child. So this is how we look at it, with this sort of the overview of environmental exposure having the gestational and the neuroimmune, affecting the neuroimmune interaction here in the center, but also direct exposure postnatally on brain development and postnatally on immune system development, because these systems continue to develop. So these early life exposures, as Bruce talked about, are really important. You have a lot of hand-mouth contact, and remembering that the body burden is so much higher because you have a smaller body mass with the same exposure that an adult is getting. That same kind of, and a lot of adipose tissue in an infant, right? They're nice and plump little, they've got all that energy store, so they've got a lot of places for those to bioaccumulate. But really looking at them, instead of looking at them as independently developing systems, now we're trying to look at them, and it's sort of the neuroimmune. And the neuroimmunology is probably the largest growing field that I know of, at least in my area. We're actually building programs in neuroimmunology because those two systems are so related. So one example of a relevant exposure that now has been banned, but as we know, because persistent organic pollutants, that's exactly what that means. They are persistent in the environment, is the polybrominated diphenyl ethers that we're looking at. Now we've got the new one to consider, which sounds like a grill to me. It sounds like some kind of the name of that, whatever that is, the fire, whatever, 550 fire. It sounds like it should be a grill. But, we sort of have, we've got these are still in the environment. So this is one of the ones that we were looking at. And we originally started looking at BD47, that congener, because that is the highest represented in that mix of compounds. That's what we see the most if we take samples. And we found really early on that there was an increase in a couple of the inflammatory cytokines in the ASD population when we took the cells from a child, expose them ex vivo in a culture dish to BD49 at 100 and 500 nanomolar levels, and then assayed for cytokine production. So this was really back in 2009. We were starting to say, okay, we're going to look at this. And then, because I work with Isaac Pesa, and I see I've got a typo up there. I don't know how 47 got into my cortical neurons there, but in the word, but where we started looking, the top panel here is vehicle control, and then neurons exposed to 10 micromolar BD47. And there's some effect, but not really profound until you go down to, this is with BD49 at 5 micromolar and BD49 at 10 micromolar. So Isaac Pesa came to me and said, okay, let's not stop with the 47. 49 is way more active neurotoxically, toxicologically, and so let's look at that. So that's what we did. So we built this study this is from the CHARGE study that we have ongoing with Irva Herz-Picciotto at UC Davis. And this is actually a fairly small study because we were doing a lot with the cells. So this was, we had a total of 98 total subjects in this, 38 with autism and 60 typically developing controls. And we did a number of measures, and this is just the breakdown of their ages and the male-female ratio. But our approach was one of the approaches is we took isolated PBMCs, so we get the blood from the kids, and we did a pre-incubation with either media or DMSO as the vehicle control, and then 50 nanomolar or 250 nanomolar BD49. We incubate them for 4 hours, and then we plate them out, and we expose them to LPS, which is an innate immune response stimulation mitogen, or PHA, which is a T-cell mitogen. And then after 48 hours, we collect the supernatants, and then we analyze them by Luminex. And at the same time, we're also measuring PBD levels and the different congeners in the plasma. So the interesting thing, I think, one of the first thing is there's absolutely no difference in body burden between the ASD population and the TD, typically developing kids. No difference at all in the body. BD47 showed up as the highest, most represented, as we anticipated that it would. But there's no difference in the levels. So there's no population difference. And then, so we took, as our measure of total body burden, then we took the highest represented, which was 47, 99, 100, 153, and 154, to determine the total body burden that we then used for the rest of the analysis against the immune aspects. But what we did notice, so this is baseline levels media. This is absolutely no stimulation. We've done nothing to the cells except they've sat in a plate for 48 hours. So this is this represents what the cells look like, just, what they're producing at their daily, sort of their baseline levels. And the first thing we noticed was that so these are all T-cell cytokines, IL-2, interferon gamma, and IL-13, was that they negatively correlate in the autism group with body burden. So that the higher the body burden, total body burden, plasma body burden of PBDEs, the lower their immune response. And then we looked at the typically developing, it was either unaffected or went the opposite direction. So now the interesting thing for us is that if you were looking at exposure, just exposure, you would not even look any further, right? Because there's no difference in the two populations. So you would say, well, BD47 or PBDE, total PBDE body burden has nothing to do with outcome, right, of autism versus typically developing. But when you look at them at the cellular level, then you actually see these really different effects. And so there's a differential response of some of these kids with autism to this, having this body burden, this exposure. And then when we looked, so that's just against the total body burden. If we looked at their ex vivo exposure, so now we're putting BD49 in the culture dish with the cells, and then we're measuring 48 hours later their cytokine production. We have the opposite. We have elevated IL-6, IL-1 alpha, and MIP-1 alpha, TNF alpha, IL-1 beta, and MCP-1. These are all inflammatory cytokines and chemokines. So now in the culture dish, when we expose them directly to this, we get a very dichotomous response. Again, the typically developing, they go down, the ASD, they go up. So they've got this sort of baseline sensitivity where they're not responsive, and then when you hit them with that congener, they're elevated. I'm trying to remember, did I put this in? So, and I was trying to keep this short, but the interesting thing is if you map that against body burden, the higher level of cytokine production tracks with the body burden of BD49. But if you map it against BD47, it does not. So it's very congener specific it's almost like, it's like a recall response. If this was an antigen, I would say, yes, they have a good response to tetanus. I put tetanus in there, they respond to it. And so this is sort of actually early data, so we will dive into this further, but there's some very specific relationship between the amount that they have in their system and the way they respond to it in the culture dish when you specifically stimulate them with it. And thank you for bringing up the Treg cells, because now I don't have to explain what they are. So I can just talk about the data. So we're trying to understand, why is this difference there? What is creating this really hyper responsiveness to this congener that was actually surpassed anything we saw with BD47, frankly. BD49 was much more active, and thank you, Isaac, for pushing us that direction. But I think the interesting thing was when we looked at the FOXP3 methylation of one of the FOXP3 promoter, so we did total change in methylation, and these are cells that came from that culture dish. So they've been stimulated, they've been exposed to the BD49. We've compared their media versus their stimulation levels, and so we took the supernames off and gave the cells to Janine LaSalle, and her graduate student looked at methylation. And what we found was that the methylation, there's a large difference, and this is so few. This is 6. We've done, because this was 6 ASD and 4 TD, because this was preliminary data to see if we could even do this. And we still got this kind of difference where there's a very rapid sort of change in, so they demethylate in the typically developing, and they don't change or they are elevated. They start out really similarly at almost the same, but there's no change in methylation with this exposure. And this corresponded to an increased production of the regulatory cytokine IL-10 in the typically developing population, where we saw a decrease in the autism population. So now we have an exposure that when we directly expose the regulatory T cells in a culture dish to this congener, we get the change in methylation happens like it should in the typically developing population and does not happen in the autism population. So this balance, I mean, Tregs are there to balance the immune response, which is what the little graphic is there for. But so now this also corresponded to the increase in the inflammatory cytokines that we saw. So we've got 2 of us working on this. So we're starting to see a pattern here of that these external exposures are, the inflammation that we see could be to issues with methylation in the regulatory T cells. And so we too will be continuing this. We've got a bunch more that we're doing now. So, this work provides the basis for exploring differential immune sensitivity, I think, to various exposures. And I think it goes beyond body burden. It goes beyond just, are they there in different levels? Obviously, that wasn't a sensitive, it's not sensitive enough for looking at what we're looking at. You need to go a little deeper, which is an expensive thing to do, and you have to pick, what am I going to look at? So, I mean, that's the difficult part of that. You have to make a choice of what you're going to explore at that deeper level. That we think that this dysregulation and sort of the hyperimmune response when we directly expose the cells has to do with Treg FOXP3 methylation and a lack of or sort of an inability to demethylate for whatever reason after that exposure. So I think these early data suggest that BD49 definitely affects immune cells from children with autism differently. Not all kids, but it absolutely related to if we looked at which kids responded with elevated inflammatory cytokines, they were the ones that were not demethylated at the FOXP3 gene. So I think we will continue to look at that. And so now I'm going to switch a little bit. I think Mark asked me to include this as well in this presentation, but maternal autoantibodies in autism is something that we've looked at for a long time. And anytime you make autoantibodies, it's because you have immune dysregulation, right? You've overcome your tolerance, your self-tolerance. You've overcome your immune regulation capabilities and pushed yourself past that regulation to create autoantibodies. And so this is something, there's a number of us that who've worked on this now, so we're not the only lab doing it. And it began for us with this pattern that you see here. If we take monkey fetal brain and we run it, actually this is human fetal brain that we did really early on. If you run it in a gel and you took plasma from mothers who had children with autism or had children with typical development or had children with developmental delay without autism, which is the DD group there, and we saw this pattern of antibody reactivity. I want to lose my mouse there. When we saw a band up at around 73 kilodaltons and one down around 37, those mothers had children with autism, and the original paper was about 12%, so that's the 2007. Harvey Singer and Andy Zimmerman at Johns Hopkins, this is the second blot down here, found something similar using a really different, they did everything very differently than we did, but also found these elevated antibodies. And then another group at the Feinstein in New York is more recently also done it, but they did it by tissue screening. So I think we've got a number of us thinking that in autism there is something in mothers during that these antibodies are present during gestation at a higher level. So the model really is that we don't know what the trigger is, so I just put that in the slide because I have absolutely no idea. There are actually 7 proteins associated with this now that we've associated. Antibodies during gestation, IgG is really important in protecting the fetus because it crosses the placenta starting around day 100 of gestation to protect the fetus from whatever the mother's been exposed to. Now, unfortunately, if you have pathologic antibodies, those are going to go in as well. And the SLE, systemic lupus, congenital heart block, is the perfect example of an outcome like that. The developing blood-brain barrier is permissive at this point, and so while it's there, it is allowing antibodies to cross where it does not when it's mature without severe provocation. But these antibodies have access to that tissue, and we now know that this IgG interferes with CNS development, and I'll give you some data on that. Increases the risk for autism, and so it's about 23% of cases, 20 to 23% that we see these autoantibodies. So we call this MAR autism, or maternal autoantibody-related autism. And a number of studies have gone into this now, and I'm not going to go through everything here in great detail because I'll run out of time, but I should have not animated this. But we know there's more severe behavior associated with these antibodies. So these are the kids, these are not the Asperger's kids, these are the kids with little or no expressive language. These are the kids with very severe stereotypic behaviors. We have a gene, the MET gene, the MET polymorphism that Dan Campbell and Pat Levitt found associated in kids with autism, we found related to the presence of these autoantibodies and because of its immune regulatory capacity. We found enlarged brain volume in male children prenatally exposed, so these are the largest brain boys come from these. And then the antigenic targets have been identified. I'll talk about those in a minute. We've got several animal models, some which we passively transferred, meaning we took IgG from a woman who had these antibodies and from a control mom, put them into a mouse, a couple of mouse studies and 2 monkey studies during gestation, and then track the offspring. But we also see both the monkey and the mouse models have enlarged brain as well. So there's a physiologic effect of these antibodies. Past that. Actually, go through. I guess this was all I had in here for this. So we are looking at the brain tissues to see histologically what's going on in them. I think one of the impacts for this is if we can identify these kids, we can get them into treatment earlier. So even if we test the mom postnatally, we can actually figure out which kids are going to go down the autism track and get them into the early intervention sooner. We could screen women at risk and develop preventative strategies. I have a grant that I'm doing that now to develop that, to be able to block it, and to define the pathophysiology. And that's one of the things we know. These antibodies target very early neurons in the developing brain, and so it's actually as the neurons are developing, not the microglial cells, not astroglial oligodendrocytes. It's neuron specific, and it really changes their developmental trajectory and how they migrate in the developing brain. So potential mechanisms. So if we think about, the things that affect, I talked about methylation and Treg cells. If you're changing the immune system in the mother and you're changing your ability to regulate the immune system, I didn't talk about all the data we have on increased elevated cytokines and inflammation during gestation and how that relates to autism, as well as how these autoantibodies, we've also found they're correlated with obesity and metabolic syndrome during pregnancy. So the presence of these antibodies is highly associated with that because that's an inflammatory disorder as well. So that susceptibility and propensity for having inflammation during gestation and how that changes developmental trajectory, one of the things that we can look at is how does diet impact the response to exposure. So as we know, denamethylation is critical in the epigenetic effects of environmental exposures, and we have sort of all the input of, these different dietary input, and then sort of how denamethylation impacts our response to stress, and then higher genomic instability, and also, and what I look at it is sort of growth, inflammation, and immune reaction. So, methylation, as we looked at in terms of the FOXP3 gene. But Rebecca Schmidt, my colleague at UC Davis, is looking at gestational folate and risk for autism, because folate is a methyl donor, and the potential mechanisms. I have to remember not to click this. So this was set up for something else. But, the B vitamins and folate and nutrients. So we're looking at nutrient status, environmental exposures. We're looking at air pollution, pesticides, and the flame retardants. And does higher intake of folate at conception and during gestation have any protective effect, and because of its role in methylation? So one of the outcomes of her study was that, and this is the tricky part, and this is when we have to go out and we talk to the community. This is prior to conception and early, at just the very early time. Well, many people get pregnant and don't know they've gotten pregnant. So, how do we, so should we have everybody on a different level of folate, because just in childbearing years, so that they are protected when they need to be protected? But there was a difference in the rate of autism in when folate was on board in that early time period, and that's really where the biggest difference was. But the interesting thing, I think, really was that folate reduces the odds of having a child by lowering the risk from the environment. So the risk is much lower when you can see all the different things that they looked at here. This came from a movie that I created, sorry. But that you can see the shift in the odds ratio with the exposure and with high folate versus low folate during gestation. So I think it's sort of an early look at how this protective, this type of thing can protect. So just, to look at the overall here, so I just talked about diet and exposure. Then we have the T cells I talked about, that the BD49, the T cells were particularly susceptible. Interestingly, BD47, it was the innate immune cells. For BD49, it seems to be more active in the T cell compartment. And these are lipophilic, which means macrophages take them up very nicely. And one of the, some of the questions we're left with, is it altering the cellular machinery? What is it doing? I mean, how is it acting in these cells? And so that's something I'm working with Isaac Pesa and Pam Lyon on. What are the methylation or demethylation defects? Why are they not demethylating? Why are the kids with autism not demethylating the way they should with the same exposure? And that's, thank goodness I work with very smart people who can help answer that question. But this, in turn, changes the cytokine, chemokine profile, and these are all inflammatory. And then with the sort of Th2 cytokine there, IL-4, which is your allergy cytokine, I guess you want to call it that. And how do these change neurodevelopment? And I think one of the aspects, one of the studies I just finished with Kaiser, the EMA study, the early markers of autism studies, we saw huge differences in inflammatory cytokine levels in women during mid-gestation, when the outcome was especially autism with intellectual disability. And changing that cytokine profile is also going to enhance your capability of producing autoantibodies. So, these are the questions that we're left with. Is this pathway for us, this is what we're looking at. And what are the genes? We may never get to what the genetic susceptibility genes are, but we will try. So this is done with there's a large group of people involved in this, and, and it's a multi-investigator approach to trying to answer this very complex question. So thank you.